I want to go a little bit more into the technical details today in my part of this webinar. So Lisa already gave a fantastic presentation, and I think at this point I don't need to convince anyone that DVP is a really powerful tool for biological discovery and also clinical translation. So to go a bit more into the technical details, I'm from Fabian Koschia's lab here in Berlin at the Delbrück Center, and we put a lot of work and effort in the past months and years into democratizing and scaling up this workflow. So there's a lot of interest currently into deep visual proteomics, and ideally every lab that wants to apply the method is able to do so. So my colleague Jose has been working on a very nice manuscript called OpenDVP that explains a lot on how to start your experiment if you're new to the field, and what to consider and what are the possibilities. And then I will talk a little bit about my project that is now also on bioRxiv on how to speed up the system and how to measure more samples in as little time as possible. So for this, I teamed up with Christoph Crisp from Bruker. We work here together in Berlin at the MDC Bruker Center of Excellence for Single-Cell Omics. It's a very nice collaboration between academia and industry. It's a lot of fun, and we're learning a lot from each other, I think. And we asked ourselves, how can we measure more samples in less time without really compromising on the protein identification and the reproducibility? And something that you need to consider when you do DVP is that we need very specific microscopy slides for this, because you cannot use the LMD and just laser from a glass slide. So we have very specific membrane slides that we use. So we need to clean all of our samples through the Evotips at the end to get rid of this membrane, obviously also to get rid of detergents, but for us it's also mandatory, so we don't inject the membrane from the microscopy slides into the mass spec. So yeah, to find the workflow on how to make this possible in a high throughput fashion, we decided on collecting the tissue ROIs into the Evochip, so that we can then process the samples in the cellenONE system using a 1-hour lysis and a 1-hour digestion step, so that this entire workflow of applying buffers, incubation times, and loading them directly from the Evo 96 chip onto the Evotips just takes 2.5 hours, and you don't need to do any manual pipetting, which I'm also a huge fan of. From the mass spectrometry side, we looked into the Whisper Zoom gradients, how they perform from the 20 SPD to the 120 SPD, and measured all of this on the timsUltra AIP system. And all the data was then analyzed in Diane. So a lot of benchmarking in proteomics usually happens on HeLa Digest, but if you want to benchmark or test some conditions for tissue proteomics, you should get some tissue as input to test this. And we decided here on 2 very different tissues. So on the left side, you can see a mouse liver. On the right side, you can see a human tonsil tissue. And in this beautiful IF staining by my colleague Sonia, you can really appreciate in green that we have those B cell rich areas, the germinal centers where they interact on the outer side, where it's a bit more, where you see this pink staining with the T cells in your tonsils on the back of your throat. And we chose those tissues because they are highly structured. So when we look into the architecture, you can select a couple of cells from 1 ROI. So you need to do 1 cut in the LMD instead of collecting multiple cells. And this is much faster for the benchmarking. So for this project, we decided on collecting 4 different tissue volumes, and starting from those very small volumes of 0.04 nanoliter up to what I define as big areas, which is very little tissue material of 0.5 nanoliter. And when you look a little bit more into detail, what that actually means in terms of cell number and the input, so the smallest input that we tested of 0.04 nanoliter, and that is approximately 100 T cells here in the tonsil, and you can see and count maybe 12 to 15 hepatocyte shapes, but because we work on 5 micron sections, we're actually slicing the cells, so this corresponds to approximately 10 hepatocytes. So we benchmarked this and collected it into the Evo 96 chip. And something that most people that have not worked with DVP yet are not aware of is that we face some issues with electrostatics sometimes because of the properties of the membrane and the tissue. but collecting into the Evo96 chip, which is very, very close to our tissue specimen inside the microscope, allows us to collect really most of the tissue that we cut. So we have a collection efficiency of over 95%. And if you have a LMD in your lab, but you don't know how to collect into this chip, you can download the STL file here to print your own adapter for the lab. Now looking at the first proteomics data, you can see how we measured for different tissue sizes and the different colors represented here, from liver and tonsil tissue, with the 4 different Whisper Zoom gradients. And you can really nicely appreciate that obviously the more tissue you collect, the more proteins you identify, and the deeper is your identification. And also with longer gradients, we can identify more proteins. But we were really happy to see that, for example, here, when you look at this very small tissue area that I showed you of 10 hepatocytes, when we run them at 120 samples per day, we can still identify more than 2,000 proteins from the small shapes, and more than 3,000 for the T-cell enriched area in the tonsil. And I think you can also appreciate here that the numbers look a little bit different, and this is why we chose those 2 different tissues, because we know that they have a little bit of they have different dynamic range properties. So we know, for example, that when you look at the cumulative intensities of the top 500 proteins, this makes up 80% of the intensities in the mass spec for the liver, and only 74 for the tonsil. So you have in liver this really high abundant proteins that cover the signal of the lower abundant proteins. So this is something that you always need to consider if you want to design your own project, and that you test this out, because every tissue is a little bit different. So it's hard to make assumptions from the beginning without seeing any data. And what surprised us a lot is that when we compared these results to our own studies from 1.5 years ago, we saw that so here we also looked into human tonsil tissue, into T-cell areas, and we can double the proteome coverage at the same speed. So here we also collected the same tissue amount. So yeah, more than 4,000 proteins here, and we quantified approximately 2,000 in this study. Or if you look at the same coverage, we can increase the throughput 3 times already. And this is really the combination of this very sensitive sample preparation methods, the really good Whisper Zoom methods that we're using here, and also the super sensitive mass spectrometers. So it is nice to see how the field develops so quickly. And protein IDs is obviously not everything. So even looking at the CVs here, we can see that they are below 20%. So we have mean CVs from like 12% to approximately 16%. And we think that you can even, so the CVs are a little bit higher in the tonsil. And we think that this also reflects some of the biology. So the T cells are a bit more heterogeneous than the hepatocytes in the liver. And with this data, you can do fantastic biological analysis. So from those very small tissue areas, we can even quantify, identify, look at transcription factors. And we were happy that we covered more than half of the immune system process related proteins that are annotated in the genome. So in the next part, we also wanted to see how the mass spec methods that you choose, how they have an input or an impact on your results. And we were specifically happy to see that from those very small tissue areas of 0.04 nanoliter, in combination with the Whisper Zoom 120 samples per day, we get those very nice results. So we looked into 3 common methods for the MS acquisition. So here on the left side, you can see the standard DIA methods. And then we also looked into diagonal parseF methods and into PyD8 optimized methods, which is a Python package that you can use. You load in your sample and you have an optimized window set up as an output that you can then use for the mass spec. And we also compared, so top to bottom panel, the 100 millisecond accumulation in RAM time in the timsTOF instrument compared to only 75 milliseconds. And this is a lot of methods that we compared. So if you're super interested, you should have a look into the preprint. But I'm showing you here some of the results. So when we benchmark this on approximately 10 liver cells, you can really nicely appreciate that with decreased accumulation and RAM time in the TIM cell, we have a deeper protein coverage. And we were really happy with the results from this method number 2 and number 6 over here. And then when we look, this is not CVs, this is the data points per peak, we can appreciate those really high numbers for the data points per peak for the PID8 optimized method. So we continued with this method. We really liked it because it also features really reproducible quantification. That you can see also here. And this is 2 neighboring tissue sections, or like 2 neighboring sections on the same tissue, and we have a really high reproducibility here with this PID8 optimized method. And to showcase this on biological more relevant sample, we looked into this rare squamous cell carcinoma of the oral cavity, and those beautiful IF stainings are done by my colleague Sonia again. And we really wanted to use the imaging information to guide us to select different areas of interest. And again, we decided here to not collect single cells, but to go more into this ROI-based approach. You can see that here over this entire tissue section that has approximately 2 centimeters of size, we isolated approximately 200 little areas. So this approximately 10 cancer cells again. And we were able to, after the collection, to do the sample preparation and also the MS acquisition of all of those ROIs here in less than 2 days. And we were really happy to see that we, of course, we had a few outliers, but at the end we received more than 170 high quality proteomes from this very low input tissue material, and we quantified in the cancer, but also in the stroma compartment, close to 3,000 proteins. And we capture known biology, so they group in different sites in the principal component analysis. And we were also happy to see that from the 2 plates we cannot see a batch effect. And if you are interested in the biology of this head and neck squamous cell carcinoma, you should have a look into the preprint. But something that I found super interesting and very nice about this technique is that we can then project the mass spec data back onto the tissue. So here we're looking at a protein, and you can see for all of the different contours that we cut out, you can see the protein intensities, and you can do you can really combine the whole set imaging information with the proteomics data. Yes, and finally, you saw a lot of data from the Evosep ONE, but we already have access to the Evosep Ino. We have one here in the basement, and Christoph at the Brucker Center, he also has one in his basement. So we wanted to see how this actually looks like on the new LC system. So I also wanted to show you some very fresh data here. We again isolated, we're looking at this human tonsil tissue, again with some B cells and some T cell areas and some epithelium that is here stained in orange on the outside. And we isolated those small tissue areas and measured them on using this sample preparation workflow and measured them on the standard gradients on the Ino. We're really happy with the protein coverage that we can see. So you get relatively deep proteomes using the standard methods like 3040 SPD, and still with 100 samples per day, we can quantify close to 5,000 proteins from those very small tissue areas. And you can also get and quantify close to 2,000 proteins with 500 to 300 samples per day, which was really happy and surprising for me, because I think this is really paves the way for huge cohort studies that were not really feasible before using the lower throughput methodologies that we have. And when we compare the proteome from the stromal compartment to the T cell compartment here, we can see a lot of many differences on the proteome level, a lot of significant differences in this t-test comparison. And we were really happy to see that you can reflect very similar processes and see the same biology also with the 500 samples per day methods. So of course, it's always the choice what you need. Is it more about the throughput, or do you need want very deep proteome data? But still, from like only 2,000 proteins, one can do very great biological analysis. Yes, and to summarize already, just in a very quick slide, I think Deep Visual Proteomics is a really, really nice tool for biology and to get ideas about what's going on in the organism, on the tissue and the tumor, for example. And I'm happy to see that a lot of people pick up spatial proteomics, and now it's also getting faster. So this allows us to do really cohort-sized proteomics analysis now. And with that, I also want to thank my team, because I presented this, but this is all a team effort. And this is us on the beach during our lab retreat. And yeah, also thank you to the collaborators. Yeah.